be equal to the isoelectric point of the protein or amino acid. Is it clear? This is the concluding statement. You select the buffer such that the pH is either less or more than isoelectric point. Why can't you use a buffer equal to the isoelectric point pH? Why can't you use it?
Because in isoelectric point, at an isoelectric point, the net charge will be 0.
0. I told you electrophoresis, only charged molecules can move. If your molecules are not carrying a charge, they will not move when you carry out electrophoresis. So, using the buffer of a required pH is very important. So, pH is firstly required to maintain the charge of your molecule and secondly to carry the current. Okay, so these are the important points. So, what are the different buffer systems that are used? Okay, so required pH is given here. So these are the different buffer components: tris, boric acid EDTA, phosphate buffer, tris acetic acid EDTA. So these are the different buffer systems which you can use to carry out electrophoresis. So it gives you the pH range. Based on your component, you can select the pH range and the buffers. Okay. So now let's study about some history of this electrophoresis. Then we'll move on to the types of electrophoresis. So this technique was introduced in 1948 by Arndt Tiselius. Okay. So this began as a moving boundary electrophoresis. Now we use paper, and mostly not even paper. Now we use either agarose gel or polyacrylamide gel, which is more stable. But initially, how was this? Let us see, it was a moving boundary electrophoresis where they used a U-shaped tube and they filled it with a buffer solution, and which was connected to a anode and a cathode. You can observe that the buffer system with a U-shaped tube is connected to an anode and a cathode, positive charge and negative charge. And now they loaded the sample on one side. Now this was a liquid completely. Okay, when you load the test sample, it starts moving based on the charge. Okay, if it is positive, it will move towards cathode. If it's negative, it will move towards anode. So this was how initially the technique was developed. But what are the disadvantages of this technique? Since it is completely only a buffer solution, the sample will start diffusing. Apart from moving because of the applied electric current, they will start diffusing because it's just a liquid medium. And secondly, the electric current will lead to convection currents where the disturbance is observed. Okay, so the molecules will start moving based on the size or charge, but there are two disadvantages. As I said, one is there can be diffusion and the other is there can be electric current, which is known as convection, electric convection. So it was not very stable. The molecules loaded were diffusing and there was a lot of disturbance due to the convection current. So what they did was looking for a stable medium on which you can load your sample so that your samples cannot diffuse or the current convections cannot disturb them. So then they came up with the paper as a supporting material. So they saw that some porous structured material can be useful to hold the sample and to maintain a stable sample. What they did, they started using paper and then they started using cellulose acetate, and after that they started using gel. So since paper and cellulose acetate, you cannot alter the size of the pores, they started looking for other alternatives where you can change the size of the pores of the supporting material so that you can separate your molecules based on your size. If you want to separate smaller molecules, you can use smaller pore size. If you want to separate larger molecules, you can use larger pore size. So then they came up with starch, and then they came up with the gels, okay, where they used agarose and polyacrylamide gels, so that the advantage is the sample loaded is more stable, they do not diffuse, and the convection currents cannot disturb them. Okay, so this is how the invention of this technique was, by using the moving boundary electrophoresis, but later it was the paper electrophoresis and the gel electrophoresis which are followed routinely. Okay, now let us study the paper electrophoresis. So our necessity here is two electrodes, positive and negative charge. So what they did was, they had two tanks, one carrying the buffer and the other also carrying the same buffer, but one is the positive electrode and the other is the negative electrode. But now these two buffers should be connected. So you had a paper, they took a paper on which the sample was loaded, either your amino acid or your protein or nucleic acid. They loaded the sample here on the paper, but now the paper should be connected to the buffer system, where one is towards the positive electrode and the other is towards the negative electrode. So what they did was, they dipped the paper initially in the buffer, a wet paper, and a connecting wicks. These are also filter papers which are connecting them to the two buffer tanks. So this is the paper on which you are loading your sample, and these two papers are connecting to the buffer. So obviously when you are dipping a paper in the buffer, it will be wet always. Do you agree, yes or no? Even this part is not dipped. By capillary action, the buffer will rise and it will get wet. Yes or no? Do you agree?
Yes, ma'am.
You
Yes,
try
ma'am.
dipping one end of a filter paper to a liquid. Will it become wet to the other end which is not dipped? Have you observed any time? Slowly it will start rising, right?
Yes.
Yeah, the
Yes, ma'am.
same way, though this part is not dipped in the buffer, your buffer can move here. So now you have placed your amino acid, proteins or nucleic acid, whatever it is. So your buffer will start moving due to the electric current. When you switch on the electrode, apply voltage, the buffer will start moving because of the ions present in the buffer. Okay. So now, if the molecules are positively charged, they will start moving towards the cathode. If the molecules are negatively charged, they will start moving towards the anode. Okay, so they will get separated. So larger molecules will move slowly, smaller molecules will run faster, and the molecules which carry more charge will run faster when compared to the molecules carrying less charge. So what are the two parameters deciding the movement of the molecule? Based on
Size and the amount of the
charge.
charge.
Yeah, based on the charge and the size, they will start moving. So they will get separated. So you have a mixture of proteins, a mixture of amino acids. Each amino acid is different in their side chain. They're different in their size. They're different in their charge. So they will start separating based on their size and charge. And you can spray them later. After they get separated, you can take this paper, spray it with a reagent which can detect amino acid and see the separation. Okay, which is the reagent which can be used to detect amino acid? I told you to check in the last class.
Then
Ninhydrin.
ninhydrin solution.
Ninhydrin is one. So you take this paper after this electrophoresis is over, spray it with ninhydrin. You see the spots wherever amino acids are present. You can see the spots. Okay, so this is the analytical technique which you can use to separate a mixture of components and analyze them. Okay. So the next, so since paper also was not more stable, they felt, and one disadvantage with paper was, even though they were stable, the disadvantage was they cannot alter the pore size. The pores were same. They wanted to separate the molecules based on the pore size. So then they started using the gels. Okay, so the gel electrophoresis came into picture, where you can alter the size of the pores, so you can allow these smaller molecules or larger molecules to be separated. So what they used, starch was initially used. Later they didn't use starch since they were not stable in all pH, and they were also not stable at the ions or where the as pH. So then they started using agarose and polyacrylamide. Okay, so now the important electrophoresis technique which are used is agarose gel electrophoresis and polyacrylamide gel electrophoresis. So how many of you have seen this type of gel with the DNA on it appearing as an orange color band? Anyone? No? So this is how the gel, agarose gel will look. So this is under UV or observing your DNA. So I'll explain the technique and I'll show you a video also so you can understand it better. So now what happens, instead of paper, they started using agarose, the polysaccharide. What do you mean by a polysaccharide? What are the building blocks of polysaccharide?
Amylose and amylose.
Monosaccharides and amino.
Yeah, monosaccharides. So monosaccharide
Monoacids.
is a single sugar unit. Disaccharide is it has two monosaccharides. Polysaccharide is it has multiple monosaccharides linked.
Multiple numbers.
Yeah, example you would have studied starch, which has multiple glucose units linked together, right? And you would have seen cellulose, also glucose units linked together, which is beta glucose, whereas starch is alpha glucose. Like that, agarose is a polysaccharide which is made up of galactose and 3,6-anhydrogalactose. So the repeating unit of agarose is galactose and 3,6-anhydrogalactose. So this polysaccharide has the ability to form a gel. So you would have seen, even while cooking, you would have seen the gelatin, how it is used for preparation of jellies. When you heat it, it will get dissolved, and when you cool, it will solidify to form a jelly kind of structure. It is same way here. Your agarose, when you heat it, okay, you can, it's available in the powder form. You can dissolve it in buffer and you can 70 to 80 degree, you can just boil it. When you start boiling either in a microwave or by using even a burner, you can boil. When you start boiling, it will dissolve. Okay, after it dissolves, you have to allow it to cool. And when you cool it to room temperature, it will form a solid. Very similar to how you make jellies, it will form a solid gel like this. But it's not a hard structure, it's a semi-liquid kind of structure where the molecules can move. Okay, so you can form a gel like this and you can load your samples in that. Okay, so that's how agarose came to use. And this gel structure, you can alter the pore size. Now, usually they use the agarose from 1% to 3%. Can anyone tell me how can you prepare a 1% solution? Okay, how can you prepare 1% agarose solution? Can you tell me what do you mean by 1% agarose solution? Do you know how to prepare 1%? Okay, can you tell me what is 1% in terms of grams? How many grams you will take for 1%? How many grams you will take if it is 3%?
Ma'am, is it 1 gram to
Yeah, 1
100
gram
ml?
in 100 ml, correct. So how do you prepare usually 1% or 3% solution is percent represents 100 ml, and that 1 or 3 will tell you how much of that solid substance you are dissolving. So if I say 1%, I am dissolving 1 gram of agarose in 100 ml of buffer. If I say 3%, I am dissolving 3 grams of agarose in 100 ml of buffer. Okay, understood now. 1 to 3%. Now tell me, I told you where agarose concentration is less here and agarose concentration is more here. So now, which will form a larger pore size gel and which will form a gel with a smaller pore size? Can you guess? Agarose is less here, agarose is more here. So 1% and 3%. Which gel will have smaller pores? Which gel will have larger pores?
3% will have a smaller this one,
Yeah,
madam.
because more
1% will have a large.
more cross-linking. Very good. So this will have small pore size. 1% will have large pore size. So you can select the percentage based on the size of your molecule which you want to separate. If your molecule is small, and then you can select around 1% gel. If your, sorry, 3% gel. If your molecules are larger, you can use gels of lower percentage. So now you know how to select the pore size of the agarose gel based on the size or the molecular weight. Is it clear? So now, how is the molecule getting separated based on the size? Now when you're loading your sample on the gel, okay, you can observe here, you will have wells here, you load your sample and you electrophorus. I'll show you the video now, and the molecules will start, and nucleic acids are negatively charged, so they will start moving, and when they move, it's based on their molecular weight. Smaller molecules will move faster and larger molecules will move slower. Okay, so smaller molecules will move faster and larger molecules will move slower. Okay, I'll show you a video now. So you just observe how this agarose gel electrophoresis is carried out.
The casting and running of agarose gels is an important aspect of molecular biology, allowing quick analysis and occasionally purification of nucleic acids. To cast an agarose gel, you will need tape, a casting tray and gel comb, a conical flask, measuring cylinder, heat-proof gloves, gel running buffer, and molecular grade agarose. Do not confuse agarose with agar, which is used to prepare culture plates. Depending on your protocol, you may also need a fluorescent intercalating dye. You will need to weigh an appropriate amount of agarose into your conical flask. This will depend on the percentage gel you require and the volume of the casting tray. A standard 10 cm casting tray will require 60 ml of gel. To determine the mass of agarose required, divide the percentage of the gel by 100 and multiply by the volume. For example, for a 0.8% gel of 60 ml volume, you would require 0.48 grams of agarose. After you finish weighing chemicals at the balance, please ensure that you clean up. Wipe the balance and the surrounding area with a damp tissue. Add the appropriate volume of running buffer to the agarose in the conical flask, in this case 60 ml. Then swell the flask. Microwave the flask on high for 30 seconds. Keep a close eye on the flask to ensure it does not overboil. Periodically stop the microwave and while wearing heat-proof gloves, swirl the flask. Initially the agarose granules will appear as small, translucent lenses. Keep microwaving and periodically swirling until these disappear and the agarose is clear. While the agarose is cooling, you can prepare the casting tray. Apply tape to each end of the casting tray, pressing the tape down firmly on all sides. Place the casting tray on a level surface. Let the agarose cool to approximately 60 degrees. As a rough guide, this is the temperature where you can hold the flask comfortably while wearing your latex or nitrile gloves. You can speed up the cooling by holding the flask under running water. At this stage, you can add your fluorescent dye if required. Remember to swirl the flask. Pour the agarose into the casting tray and insert the comb. Inspect the gel for air bubbles, particularly around the comb. Down can be either popped with a clean dry pipette tip or moved to the ends of the gel. Allow 30 to 40 minutes for the gel to set. It should appear slightly opaque once it's set. Once set, remove the tape and place the gel in the gel tank. Remember, nucleic acids are negatively charged and will migrate towards the positive. Therefore, the comb should be located towards the black cathode end. Pour the running buffer into the gel tank to just cover the gel.
I told you usually anode will be red in color and cathode will be black in color, okay? So these are the two electrodes.
The buffer level should be just a few millimeters above the tops of the wells. Carefully remove the comb. The gel is now ready to load. Often it can help to see the wells if there is a dark surface beneath the gel tank. Load your samples onto the gel. Try to get into a comfortable position and if possible, support your pipetting arm. Pipette slowly to allow the denser samples to fill up the wells. Ensure you leave a space for the size markers and record which lanes contain which samples. Place the cover on the gel tank and plug it into the power supply. Set the appropriate voltage on the power supply and begin the run. You should be able to observe current on the power supply. And also see bubble formation on the electrodes. When the run has finished, turn off the power supply and switch off at the mains. You can then unplug the gel tank and remove the gel. Hold the gel tray by the ends to prevent the gel from slipping off the tray. You are now ready to look at your gel under the transilluminator.
Okay, did you all observe? So that is how the electrophoresis unit for agarose gel looks like. So we call it as horizontal gel electrophoresis unit. Okay, it moves from right to left or left to right, right? Horizontally it moves. So usually for agarose gel electrophoresis, we use the horizontal gels, and you saw how the gel was casted, dissolving agarose, heating and solidifying, and you placed like a comb to make wells where you can load your sample. That is where you can load your DNA or RNA. And when you had observed, did you observe what was the color of the sample which was loaded into the wells?
Blue, ma'am.
Blue.
Purplish blue.
Purplish blue. Why? Because you know that nucleic acid solutions are colorless. And how will you know whether it's moving or not? So what they do is they add a tracking dye or a blue color dye known as bromophenol blue. So they'll mix your nucleic acid with bromophenol blue and glycerol. Why glycerol? Because you know glycerol is more dense. So when you load it in that buffer, it should not come out again if you're not making it denser. So they add glycerol to your sample so that sample becomes dense and it will settle down when you're loading the sample placed in the buffer. So no, you should know that it will run immersed in buffer. You saw that, right? So to give the density and to give the blue color, you can mix the sample of your proteins or nucleic acids, whatever it is, with the dye bromophenol blue and glycerol, and you will load it. And then when you connect the electrodes, it will start running, and you can see the movement based on the blue color dye, right? The blue color dye will start moving. So when it is reaching the end, you can stop the electrophoresis. Now, how can you visualize? For DNA, we add what is known as ethidium bromide. Okay, when you prepare the gel, you would have observed initially they added some component to the gel which is known as ethidium bromide. So this ethidium bromide is a molecule or a chemical which reacts with nucleic acids and will give orange color fluorescence under UV light. So when you observe the gel under UV light, it will give orange color fluorescence. But when you see the gel like that, normally under any light, nothing you can see. The gel will look blank. You have to observe for DNA only under UV light. You will have a cabinet. You can place your gel there, and when you observe under the UV light, you can see it will appear like this. Orange color bands will appear, which will show that DNA is there and the nucleic acids are separated, and you can check the molecular weight by comparing it with a ladder. So you can load a ladder which shows 10 base pairs, 20 base pairs, like that. Molecular weight of nucleic acid is represented as base pairs. You can run that, load your sample and check the weight size of your sample by comparing with a marker or a ladder. If it is proteins, you have to stain the gel by keeping it in Kumasi blue. I'll show you one here. This is for page, but even for agarose, it will appear like this. When you put the gel in Kumasi blue, wherever proteins are present, you will see blue color bands like this when you have proteins. For DNA, you will use ethidium bromide, which will be given a orange color fluorescence. Now, is it clear to everyone, agarose gel electrophoresis, how to
Yes, ma'am.
prepare the gel, how to pour it in the unit? You would have observed it. So practically you know how it is done. So weigh agarose, dissolve it, heat it, pour it, solidify, then load your sample with dye, bromophenol blue and glycerol, let it run. Then you take it and observe under UV for DNA or RNA. For proteins, you can stain it with Humasi blue. Is this clear? Any doubts in this agarose gel electrophoresis? Any doubts? Can we move on to the next electrophoresis technique?
Yes, ma'am.
Electrophoresis technique routinely used is polyacrylamide gel electrophoresis. Okay, so this gel came into when starch was not very stable and it started reacting and it was degraded by change in pH on all those disadvantages were there. So to get a inert gel and to get a varied pore size apart from agarose, they also started using this polyacrylamide gel. So like agarose, how is this polyacrylamide gel? It's not very simple like agarose where they used only one component, where you added agarose, heated and then you allowed it to cool and it solidified. But polyacrylamide is not like that. Let us see what are the components and solidify it to form a gel. It's little different. In case of polyacrylamide gel, you use components like acrylamide, bisacrylamide, also known as N-prime methylene bisacrylamide, and then you will use TEMED, tetramethyl ethylene diamide, and ammonium persulfate. Okay, so what are the components which you use? Acrylamide, bisacrylamide, TEMED and ammonium persulfate. What will happen now? You don't have to heat and cool and all those techniques here. If you add the TEMED and ammonium persulfate to acrylamide and bisacrylamide, this ammonium persulfate will initiate a polymerization reaction. Okay, it will form a free radical. In presence of TEMED, ammonium persulfate will form a free radical and it will start joining this acrylamide and bisacrylamide. Otherwise, acrylamide and bisacrylamide cannot join. But when you add TEMED and ammonium persulfate, they will start linking acrylamide and bisacrylamide molecule. So when they start linking, they will form a polymer and solidify to form a gel-like substance. Is it clear to everyone? So apart from TEMED, you can also use a light to initiate a polymerization in presence of riboflavin. So these are the two ways by which you can lead to polymerization of acrylamide and bisacrylamide. For acrylamide, the gel is run in a vertical direction. You saw that for agarose it was in the horizontal direction. But for polyacrylamide they prefer vertical direction from top to bottom. They will allow the movement of buffer and the sample from top to bottom. But for horizontal agarose gel it was sideways, horizontally, right? So this is how the polyacrylamide setup looks. Okay, so now I'll just explain the two different techniques which can be carried out by using polyacrylamide gel. You can carry out native phase and yes. What? You're not able to see the PPT? Others?
It's
We can see,
visible,
ma'am.
ma'am.
Okay, you try to refresh. Whoever is not able to see, just try to refresh it once. Okay, so in case of this also forms a gel very similar to agarose, but it will be very thin and it is more delicate when compared to agarose. It's not so thick as you can see. Like if I say hemoglobin is a protein with 4 subunits, meaning 4 polypeptide chains will be linked together. When you treat it with SDS, beta mercaptoethanol, and when you heat it, the 4 subunits will get separated and you will get 4 separate bands like this. Now here this protein has 2 subunits. So when you treat it with SDS and beta mercaptoethanol, you see heating is separating these two in presence of SDS and beta mercaptoethanol, and 2 bands you will get based on the size. So this is a smaller polypeptide chain, this is a larger polypeptide chain. And when you subject it to acrylamide gel electrophoresis, they will start moving. Now all of them are negatively charged because SDS is binding to it. So separation is based only on the size. So the smaller size will run faster and the larger size polypeptide chain will run slowly. Okay, you can observe this by staining with Kumasi blue as I told you. Whereas what is happening here, in this protein you have just one polypeptide chain. So you don't have the subunits formed here, just one polypeptide chain. Your SDS will go and bind to it, make it negatively charged so that it will start running. So how many bands are you getting here? How many bands are you getting
Three.
here?
Only
Three.
One
ABC.
one.
If your
Band.
protein has one subunit, you will get one band. In this case, your protein has two subunit. This is one subunit, this is the other subunit. So they will get separated when you treat it with beta mercaptoethanol and SDS. So how many bands are you getting here?
Two.
Two subunits. Okay, now one more thing to think now. Just I'm telling you, your hemoglobin has 4 subunits. Okay, but 2 subunits are having same size and other 2 subunits are having same size. So you take hemoglobin, it has 2 alpha subunits and 2 beta subunits. And the size of alpha subunits are same and the size of beta subunits are same. Now think and answer me, when you treat hemoglobin with SDS mercaptoethanol and then when you run in polyacrylamide gel electrophoresis and then you stain with Kumasi brilliant blue, how many bands will you get? Hemoglobin.
It should be 4.
4. 2 and 2.
Four. Yes.
4 subunits it has, but what did I tell you? 2
I think it formed two bands.
Sir,
Two
2
bands.
bands.
2 bands only, because this
They're
separation
similar
is
to-
based on the size. So since 2 bands are of same size, it cannot distinguish between the 2 band which is of same size and molecular weight. So you can determine the subunits given that the given subunits are of different molecular weight. So it shows that hemoglobin will show only 2 bands because 2 subunits has same molecular weight and the other subunit will have same molecular weight. So by doing this SDS page, what all you can determine is, you can determine the molecular weight by comparing it with standards. You can run standard protein of known size and molecular weight, and then you can compare your unknown sample with your sample which is known molecular weight. You can plot a graph and then determine the molecular weight. And the other is, you can determine the subunits. How many subunits does it have? That also you can determine. As I shown here, this will show it has one subunit. This will show it has 2 subunits. But further, are there subunits of same molecular weight? You can do analysis by doing other techniques. Okay, so this can set the basis on which the molecule separate is only the size and molecular weight. Because charge is uniform, so the separation is based only on the molecular weight and size. Smaller molecules will run faster, larger molecules will run slower. Or I can say less molecular weight compounds will move faster, less molecular weight substances will run slower. Have you understood this SDS page now? Is it clear? Okay, now let us see what is native page. So it's the same polyacrylamide gel electrophoresis where you cast the gel here, acrylamide, bisacrylamide, take APS temid, polymerize, load the sample, allow it to run, and you can stain and observe the bands. But what do you do in native pages? You don't add this SDS or beta mercaptoethanol. So we say it is a non-denaturing gel. This SDS page is a denaturing gel because it's leading to denaturation where your fragments are getting separated. So it is a denaturing gel electrophoresis. Whereas in native page, you will not use SDS or beta mercaptoethanol. It is a non-denaturing gel. Now tell me, when you carry out a native page where there is no SDS or beta mercaptoethanol, so how many bands will you get for this protein which has 2 subunits and for this protein which has 1 subunit? How many bands will you get? No SDS, no mercaptoethanol. So how many bands will you get for this protein which has 2 subunit?
Only one band,
Only one. Why?
because it's not getting denatured and the polypeptide chain is not
Denatured
cut.
and
Yes, very good. So it's
the
not
chain
getting
can't be cut.
denatured. So as a whole, it will run native. It will run in the native conformation. That's why we call it as native page. Even this will have one band because it doesn't have subunits at all. So this protein will not get separated. It will run. So you can determine the size of the complete protein in case of native page. Whereas in SDS page, it will get separated into subunits, whereas here the complete protein will run. So the protein will retain its shape, structure and its activity. Nothing will happen to your protein. Whereas in SDS page, since you are heating it, you are treating it with SDS, mercaptoethanol, the proteins are denatured, the activity of the proteins will be lost. But in native page, you can retain the activity of the proteins. Like most of your enzymes are proteins, right? Except for the ribozymes, all the enzymes are protein in nature. So you can check for the enzyme activity by running on the native page because enzyme is not denatured, it maintains its activity. But in SDS page, you cannot check the enzyme activity because it's all denatured, fragmented. So you can check what is the size and subunits of the enzyme, but you cannot see the activity of the enzyme. But in case of native page, you can run the enzyme, which is a protein, add substrate to that and see if it is reacting, and you can check whether enzyme activity is there or not. So we say that native page is also used for checking the enzyme activity, which we say as in-gel enzyme assay. Okay, so now the difference in native pages, since no SDS, separation is not only based on the size, it's based even on the charge of the molecule. In SDS page, it was uniformly bound by SDS, but in native page, no SDS. So whatever is the charge of that protein, based on that and the size, they will get separated. So just in one line, if you have to remember, in native page, native molecule gets separated under non-denaturing condition, and the separation is based on both charge and mass, charge and size. Whereas in SDS page, it is a denaturing condition are different and size is different. So separation is based on charge to mass ratio. We say that in one line, in native page, molecules get separated based on charge to mass ratio. In SDS page, molecules get separated based on their size or molecular weight. Okay, so now tell me, if you have two proteins of same size and molecular weight, but protein A has more charge and protein B has less charge, which will move faster in native page? Two protein of same size, meaning same mass and molecular weight. Protein A has more charge, protein B has less charge. In native page, which will run faster?
Protein A.
Protein A.
Protein A.
Protein
Okay,
A.
very good. So size determines its movement. It's not only the size, the charge also determines the movement in case of native page. But in SDS page, only size or molecular weight matters because SDS is binding to all the proteins uniformly. Charge does not vary. So this is the simple concept which you should know for polyacrylamide gels. Okay, which is SDS page and native page. Okay, you can see that. I'll just show you a video now to see how the polyacrylamide gel electrophoresis unit is appearing and how is it casted, how it is run. So as I showed you the picture, this is how the protein bands will appear when you stain the gel with Kumasi blue. After you run the gel, you have to put it in the solution of Kumasi blue. You can get bands wherever proteins are present. It will appear as blue bands. Okay, so now I'll share the video. All of you can see the video now.
sulfate, polyacrylamide gel electrophoresis, is an electrophoresis method and very powerful technique in the study of proteins. In SDS page, proteins are separated in a polyacrylamide gel based on their molecular weight. For sample preparation, a loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue, and glycerol is added to the protein samples. Protein samples may be biologically derived, for example from prokaryotic or eukaryotic cells, tissues, viruses, environmental samples, or purified proteins. When the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes. Proteins are large biomolecules consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain. Proteins are formed by linking amino acids with peptide bonds and fold into one or more specific spatial conformations, driven by a number of interactions such as denatures the native proteins by disturbing the hydrogen bonds, hydrophobic and ionic interactions, while the reducing agent beta-mercaptoethanol is used to cleave the disulfide bonds. Also, when a protein mixture is heated, SDS binds uniformly to the protein, and the intrinsic charges of this protein become negligible when compared to the negative charges contributed by SDS. This treatment brings the folded proteins down to linear molecules with net negative charge. Therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. For gel preparation, a separating gel solution with pH 8.8 and a stacking gel solution with pH 6.8 are used, and both consist of acrylamide, bisacrylamide, ammonium persulfate and T-med. The gel is produced by polymerization between two glass plates anchored vertically in a cassette. The separating gel is poured first. The polymerization reaction is a vinyl addition polymerization initiated by a free radical generating system. Ammonium persulfate is used as the free radical initiator, while T-med accelerates the rate of formation of free radicals from persulfate. These free radicals convert acrylamide monomers to free radicals which react with unactivated monomers to begin the polymerization chain reaction. The elongating polymer chains are randomly cross-linked by bisacrylamide, resulting in a gel with a characteristic porosity. The pore size of the gel is related to the ratio of acrylamide to bisacrylamide. Low concentration is preferred for high molecular weight samples. The stacking gel solution is poured on top of the solid separating gel, and a plastic comb placed on the top of the stacking gel during polymerization enables the formation of small wells in the gel. Once the gel completely polymerizes, the comb is removed and the gel cassette is placed vertically between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode positioned at the top of the gel. The gel is inserted into a chamber, and a tris glycine chloride buffer system with pH 8.3 is poured to allow the conduction of current through the gel. SDS is also present in the gel and in the running buffer to make sure that once the proteins are denatured, they stay that way throughout the run. For sample application, in addition to the samples, a molecular weight size marker is usually loaded onto the gel. This consists of proteins of known sizes and thereby allows the estimation of the sizes of the proteins in the actual samples, which migrate in parallel in different tracks of the gel. Each sample is pipetted into its own well in the gel. And as we have seen previously, bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is useful for visualizing the sample in the well, and glycerol increases the density of a sample and used to make the sample fall to the bottom of the sample well rather than just flow out and mix with the running buffer. After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly through the gel. The stacking gel is used to ensure that all of the proteins arrive at the separating gel at the same time. Because gel wells are around 1 centimeter deep, so in the absence of a stacking gel, proteins would all enter the separating gel at different times, resulting in very smeared bands. As we have seen previously, the running buffer consists of glycine and chloride ions. So, in the sample wells we will have proteins, chloride ions, and glycine. Proteins and chloride ions are negatively charged. Well, glycine can exist in 3 different charge states depending on the pH. The isoelectric point or pHi of glycine is equal 5.97. At this pH, glycine carries neutral charge due to the gain and loss of protons. At a pH below its pHi, glycine carries a net positive charge. And at a pH above its pHi, it becomes negatively charged. The running buffer pH is equal 8.3, which above the pHi of glycine. So, at this pH, glycine is negatively charged. Once the electric current is applied, proteins, chloride ions and glycine are forced to enter the stacking gel, where the pH is equal 6.8. At this pH, glycine switches predominantly to the zwitterionic neutrally charged state. This loss of charge causes them to move very slowly in the electric field. The chloride ions, on the other hand, move much more quickly. Chloride ions migrate in front of the proteins as leading ions, and glycine molecules migrate behind the proteins as initial trailing ions. Consequently, proteins are trapped and concentrated into a narrow zone between the chloride ions and glycine. This procession carries on until it hits the separating gel, where the pH switches to 8.8. At this pH, the glycine molecules are mostly negatively charged and can migrate much faster than the proteins. Proteins are separated, so higher molecular weight proteins move more slowly through the porous acrylamide gel than lower molecular weight proteins. Due to the relatively small molecule size of bromophenol blue, it migrates faster than proteins. And by optical control of the migrating colored band, the electrophoresis can be stopped before the dye and before the samples have completely migrated through the gel and leave it. At the end of the electrophoretic separation, all proteins are sorted by size and can then be analyzed by other methods, for example protein staining methods and immunological detection, such as the Western blot technique. After removing the glass plates, the stacking gel is discarded and the cell can be stained. Kamasi blue is the most popular protein stain. It is an anionic dye, which non-specifically binds to proteins. The structure of Kamasi blue is predominantly non-polar, and it is usually used in methanolic solution acidified with acetic acid. Proteins in the gel are fixed by acetic acid and simultaneously stained. After staining, the proteins are detected as blue bands on a clear background. The molecular weight size markers in a separate lane in the gel can be used to determine the approximate molecular mass of unknown biomolecules by comparing the distance traveled relative to the marker.
Okay, did you all watch the video? Have you all understood?
Yes, ma'am.
Have you all understood how the polyacrylamide gel electrophoresis is carried? Did you all get an idea how the apparatus looks?
Yes, ma'am.
Yes, ma'am.
Okay, so that details of that ions, it's not necessary for you to remember. Like, I'll just tell you that the gel, lower part of your gel will be resolving gel and the upper part will be stacking gel. Okay, stacking gel will allow the proteins to become compacted so that before entering into the resolving gel. So that's all based on the ions and pH of the buffer. So all those details are not required for you. So remember that SDS page, what is the function of SDS? What is the function of beta mercaptoethanol? And what is the basic principle on which they are separated? So anyone, can you tell me now what is the basic principle of SDS page on which the molecules are separated? Based on
Denaturation
no what?
and no, the bands will be based on the size.
Size. Very
Size.
good. In
Size.
native page, how is it different from SDS page? How is native page?
Charge to the native page,
Separation
it is
is
charge to the mass ratio. The charge has been
So there charge also counts. So separation
and
is
mass.
based on both charge and mass. So the apparatus looks same even for the native page. Only thing, you don't add SDS and beta mercaptoethanol to your sample and to the buffer. Whereas all the other components will remain same. You will use acrylamide, bisacrylamide, APS, TEMED for polymerization, and you'll use buffer with tris and glycine. But here you will add SDS. In native page you will not add SDS. Okay. So there's another video. That video is very long, around 38 minutes it is. I'll share you the link after the session. You can see like exactly in the lab how the gel is casted. Okay, if you have time after this, you can watch. I'll share you the link for the video. This was an animation which was shown, but that is exactly how it is done in the lab. So if I play that, half an hour will be gone. So you can look at the video later. I'll share the link later. So now I'll share the PPT. We'll continue. Okay. So we completed native page, SDS page, agarose gel electrophoresis. Now one more concept is isoelectric focusing. So this is based on the isoelectric point of protein. So what you have to remember is, I told you that proteins carries no net charge at isoelectric point. Below that they are positively charged, above that they are negatively charged. So you can run the proteins or amino acid in a gel which is ranging in a pH. So the pH of the gel is not uniform here. You can cast a gel such that the pH is ranging from low to high. For example, pH is low here. I will tell 1, 2, 3, 4 like that. It goes up to 14 for example. So the pH of the gel, if you take here, it will be 1, and if you take here, it's going to be 14. And in the middle, it will be 7. Okay, so you use a gel which is known as a isoelectric gel where the pH varies. The pH of 3, it is the isoelectric point for that protein. So what will happen now? Will it move further? Will this move further?
No.
No net charge. So it will not migrate, it will stop here. So this band is formed here. So observe here that the pink band is formed here. So the blue color molecule, the isoelectric point is here. For example, you say 5. So below they will be positively charged, above they will be negatively charged. So when they are here, they will start moving, and when they are here, they will start moving towards the negative side. Like they will move until the zwitter ions are formed. So here no net charge, it will stop here, and they will form a band here. Like this, isoelectric focusing is a technique which separates the proteins based on their isoelectric point. So when they reach the isoelectric point in the gel where there is a varying pH, they will stop moving and the bands will be formed. So they have given you an example here. So the different proteins and enzymes, these are the various enzymes and proteins, cytochrome C, ribonuclease, lectin. See what are the isoelectric point. So they will form band at the respective pH which is equal to their isoelectric point. So remember that in case of isoelectric focusing, you will have gels which has varying pH, which is formed using chemical components known as ampholites. How do you get varying pH? By using chemicals known as ampholites. You can cast a gel which is varying in pH. Then you can load your samples, and samples will be positive or negatively charged based on the pH, and they will start moving, and when they reach the isoelectric point, they will stop moving and they will form a band at that point. Is this clear now to everyone, isoelectric focusing? Yes? Is it clear? Any doubts in this isoelectric focusing? Any doubts?
No, ma'am.
Okay, so you have to remember agarose gel electrophoresis, native page, SDS page, and isoelectric focusing. Yeah. Now I have to move on to the next technique, spectrophotometry. So before going, all of you can take a 5 minutes break and come back. And this I'll take another half an hour and then we can finish this, okay? So you can take a break for 5-10 minutes and come back. Okay, can we resume? Response, please. Can we resume the session?
Yes, madam.
Yes, ma'am.
Okay, so we have finished the first part, electrophoresis. So the next part which we need to study is the spectroscopy, the basic instruments which you would be using for all the experiments. Calorimeter is the one which you would have used for most of the estimations. So based on what principle this instrument works is what we are going to study now. Okay, I'll share the PPT. So we'll see the principle, the instrumentation of the spectroscopy or spectrophotometry. So when you see the estimation of protein or carbohydrates for most of the assays, inorganic phosphate, you will add, yesterday I showed you one picture in day before yesterday's class. For the quantitative analysis, you use concentration, different concentration, and add a reagent where you get different color. And I told you you can measure the intensity of the colored samples. Do you all remember that qualitative test and quantitative test which I showed you in the last class? Yes?
Yes, ma'am.
So this is how you do the quantitative analysis. So I told you one of the quantitative analytic method is photometry, absorption photometry, which is nothing but the spectrophotometry we will be discussing today. Okay, so what do you do in this basically is to check the concentration quantitatively, to check how much of the component is present exactly in the unknown sample, you do this assay. I told you use a blank where there is no sample at all, and you use your sample at different concentration, and you take your unknown, right? And just admit. Okay. So what have you done here? This is how you do the assay. You will take samples of different and you take your unknown sample where you don't know the concentration and you add a reagent that can form a colored solution. You check the intensity of the color and compare the intensity of the unknown color with the standard and tell how much is present in your unknown. Quantitative technique which is effective, easier to follow and accurate also. So how do you measure that intensity of the color, or why do you have to make the substance to form a colored complex? So all of these we will be discussing in this. So these are the instruments where you will measure the intensity of the color. Now I showed you, right? You will have a small glass cuvette in which you will pour the sample, then place it into this and you will check the OD, which will give you the intensity of the color. Darker the color, the value which you will get will be more. Lighter the color, the value which you will get will be less. Okay, so this is the instrument known as colorimeter. Same principle it works, but it is a more sophisticated instrument which can give you more accurate and more sensitive results. Okay, I'll explain you when I come. So this is known as colorimeter. This is known as spectrophotometer. So these are the routinely used instruments. So we should see now on what principle. So now what are those in your molecules that can absorb light in the UV range or visible range? Okay, so in your proteins, you have structures known as peptide bonds. You know that proteins are nothing but amino acids linked together, right? So in your proteins, the peptide bond or the amino acids like tyrosine, tryptophan, all of them can absorb the light in the UV range. Okay, UV range is a light with the wavelength ranging from around 150 to around 330 or 340 it goes up to. So visible ranges from 400 to 700, light with a wavelength of 400 to 700. So here in colorimeter or spectrophotometer, when you have proteins, they can observe only in the UV range. But if you want your molecules to observe in the visible range, you have to make them colored. That's why we add some reagent and make them colored. For protein, you add biuret and make them colored. For amino acids, you saw that you can add ninhydrin. And for carbohydrates, what can you add to make them colored? Any guess? What reagent can you add to reducing sugars to make form a colored complex?
Benedict solution?
That is qualitative test. Here you use what is known as DNS, dinitrosalicylic acid. So when carbohydrates react with DNS, like your reducing sugars will form orange color, red color. Like that proteins react with biuret, they will form purple color. Or when amino acid reacts with ninhydrin, they will form a purple color. So you have to form a colored complex for the molecules to absorb in the visible range. So now what are we going to see is, you will place your sample in the cuvette and you will allow the light to pass through it, because you are just checking the interaction between the electromagnetic radiation and your sample. So you are placing the sample allowing the light to pass through it. Okay? In the UV range, your samples can observe even without making them colored. Like you don't have to make them a colored solution by adding a biorate reagent or by adding any other reagent. In UV range, they can directly absorb because they have this peptide bonds, tryptophan and the other structures in the proteins. So your proteins can absorb directly in the UV range without making them a colored substance. So they can absorb certain light and the remaining light will pass through. But if you are using the visible part of your light, your solution has to be colored. So how do you make it colored? By adding certain reagents. So in either cases, in UV, when you're passing the UV light, the color is not required. But when you pass the visible light, you need your sample to be colored. In both the cases, the sample will absorb light. Okay, so the light which is passing through the sample is known as incident light. Sample will absorb certain amount of light and the remaining light will be transmitted. Can you see actually it is dark here and then it's becoming lighter. Why is this blue color becoming light here? This is darker and why have it shown it light here? What is happening now? What is happening?
तो क्या दोनों सेम होते हैं
What is happening?
absorbed
Yeah, light is getting absorbed by the molecules. So the intensity of the light is decreased. So the light which is passing through your sample is incident light and the light which is coming out after your sample has absorbed is known as transmitted light. The ratio of the incident light and transmitted light is known as absorbance. That is what you are measuring in the calorimeters. The value which you get in calorimeter is showing the absorbance. How much your sample can absorb. How much of light your sample can absorb. That depends on the intensity of the color. And that intensity of the color in turn depends on the concentration of your sample. So if you have less protein and more protein, when you add biorate reagent, which will show a darker color, less protein or more protein? Which tube will show a darker color?
less protein
Which will show a dark color? You have protein.
More protein concentration.
Yeah, the protein which has more concentration will show dark color. The protein which the lesser concentration will show a lighter color. So the amount of light absorbed is dependent on the concentration. Okay, so that is what is stated by the Beer-Lambert's law, which is the principle of this spectrophotometry or UV visible spectroscopy. The principle is Beer-Lambert's law. Now what does this Beer-Lambert's law state is absorbance. Absorbance is the amount of light absorbed by your sample. So as I showed you in the previous picture, more concentration, the color will be more and the amount of light absorbed will be more and the value displayed on the calorimeter will be more. So the absorbance value which will be shown here will be more. So Beer-Lambert's law states that absorbance is directly proportional to concentration and the path length of light. So when will you get more value? Directly proportional to concentration. When concentration is more, absorbance will be more. When concentration is less, absorbance will be less. So your absorbance is directly proportional to concentration and the path length. Okay, so absorbance is directly proportional to concentration and the path length of light. So this is what the Beer-Lambert's law states. This is the principle. Okay, so when a beam of light passes through the substance, And now you have to go deeper. What do you mean by a beam of monochromatic light? Okay, so I showed you in the picture here that UV light is in the range of 150 to around 350 nanometer. Visible light is from 400 to 700 nanometer. Now, if I say monochromatic light, it is a light of single wavelength. From this light, you select a light of wavelength 400 nanometer. So it's a single wavelength. So, or 300 nanometer or 350 nanometer, whatever it is, you select a light of single wavelength. So that is known as monochromatic light. So you can't just pass a light like that by using a torchlight or anything. You have to select a monochromatic light and then allow it to pass through your sample. Okay. So you have a light source, select a monochromatic light with a single wavelength, allow it to pass through your sample and you check how much is the absorbance. Absorbance will be directly proportional to concentration. Concentration increases, absorbance increases. Concentration decreases, absorbance decreases. Okay. So this is a constant which is known as molar extinction coefficient. So this is Beer-Lambert's law, which is very important. You need to remember. It states that absorbance is directly proportional to concentration and path length. Okay, and epsilon is the molar extinction coefficient. So this is the Beer-Lambert's law, the basic principle on which the colorimeter and spectrophotometer works. Okay, now moving on to the instrumentation. Now before going into instrumentation, I'll tell you the basic differences when to use a colorimeter and when to use a spectrophotometer. So as I told you, if you have a protein solution, it can absorb in the UV range without making it into a colored solution. It can absorb in the UV range. So in that case, you use a spectrophotometer. Here you need not have a colored solution, but you can use colored solution also in spectrophotometer, but in case of colorimeter, it has to be only colored solution. You cannot measure in the UV range. Colorimeter you can measure only in the visible range. You can use only visible light for measurement, not UV light. But spectrophotometer use both UV light and visible light for measurement. You can check the absorbance by using both visible range and the UV range. Is it clear to you? First difference. Colorimeter you can measure only in the visible range. Spectrophotometer you can use both visible range and in the UV range. So in the colorimeter only colored solution can be used. In the spectrophotometer for visible range you can use colorless solution. For the UV range you can use colorless solution. Visible range you can use colored solution. This is the first difference. Okay, so now I'll move on. Let's see the instrumentation. What are the parts of this colorimeter and spectrophotometer? Both has similar parts, few differences I'll tell you. So you understood the first difference between colorimeter and spectrophotometer. Now the light source. When you take the instrumentation, the first component of your instrument is the light source. You need a light, right? You have to select a monochromatic light. So what is that which is producing a light? For the visible range, you use a tungsten filament. For UV range, you use deuterium bulb. So what is the light source? For visible range, you use a tungsten bulb. For UV range, you use a deuterium bulb. Okay, then light source. But now your aim is to select a monochromatic light. What do you mean by monochromatic light?
Single light.
Single wavelength.
Light of single wavelength. Okay. So now you have to select a light of single wavelength. So pass this light through a narrow entrance. Then you have monochromator. What is the function of this monochromator? It selects a light of single wavelength. The function of monochromator is it generates a monochromatic light, or from the light of multiple wavelengths, it just selects a light of single holder. You're placing your sample in cuvette. So this is known as a cuvette. Now the difference comes. For colorimeter, you use cuvettes made of glass. For the spectrophotometer, in the UV range, you should use cuvettes made up of quartz. Okay, quartz cuvette. You have to use quartz cuvette in the UV range. Okay, quartz cuvette, QUARTS. But for the visible range, you can use glass cuvettes, which are made up of normal glass. Understood? So what is the cuvette used in the visible range? And what is the cuvette used in the UV range?
In UV range, it is quartz.
Quartz.
In visible range, it is glass quite.
Okay. So now tell me, if you have colored solution, which cuvette you will use?
Quartz.
Quartz cuvette.
Colored solution, you will measure in the visible range.
Visible range, yes.
So which cuvette you use?
Glass.
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 use.
Then for visible UV range, you are using colorless solution. So which cuvette you will use for UV range
Quartz.
and colorless solution?
Quartz.
Quartz cuvette.
Quartz
Okay, so you should know the difference. So now you pass the light, your sample will absorb, and the transmitted light is detected by using a detector. And then this detector will display the value of absorbance on the instrument. So this is the instrumentation. Light source, monochromator, then sample holder cuvette, and then the detector. This is the basic components of both colorimeter and spectrophotometer, but I've told you the difference. Here for UV light range, which light you will use, which is the light source?
Monochromatic light. No?
Now what is the light source for UV and visible?
Deuterium or hydrogen lamp for UV light, and tungsten lamp for visible range.
Very good. Monochromator, in case of colorimeter, glass filters are used. In case of spectrophotometer, prism and grating is used.
Prism and
Cuvette,
grating.
for visible range, glass cuvette. For UV range, quartz cuvette has to be used, and you have a detector. So this is how the instrument works. So you place the sample, it will check the absorbed light, and the detector will display what is the absorbance, how much of light is absorbed, and it will display. Okay, now there's another concept, single beam spectrophotometer and double beam spectrophotometer. So in case of single beam spectrophotometer, there will be one place to keep your sample. So in this, first you will keep your blank, okay, where blank will not have any color because you're not added any sample there, right? You don't have added your protein at all. So no color there, or whatever the color of the reagents is, it will be the color of the blank. So in the instrument, okay, what you will do is, first you will place the cuvette with the blank here in this place, and then you will add this to a particular wavelength, and then you will your net color, then check the value, and then you keep on placing the next sample with increasing color concentration, and you will observe here the value will be increasing. Okay, even in this spectrophotometer, if you open this, you will have a place to keep a cuvette like this. Here it's closed, open,